I'm glad to be here to present the results of our research group from Serbia. And special thanks to Dragan and Dragica, who are on their maternity leave. So they aren't here with us, but they did a lot of research and for our research team. So today's subject is about mesenchymal stem cells and the immune cell-driven acute hepatitis. So first we'll talk about mesenchymal stem cells. These are adult stem cells that are self-renewable, like other stem cells. But what's important, they also reside in the liver and other organs, and they have a role they can modulate the immune response in the liver, and also they're important for the renewal of hepatocytes. And also after the liver damage, whether it's of infectious nature or toxic, they maintain homeostasis in the tissue, and they are important for repair and regeneration. They do this by different mechanisms. So after the elimination of the pathogens, they induce the immune response, which is important for the return to homeostasis and for the repair processes. And also they can do that by cell-to-cell contact or by secreting cytokines which have immunomodulatory properties like TGF-beta and interleukin-10, and also other substances like nitric oxide and prostaglandin E2. And all of these cytokines, they shift the immune response toward the reparation processes, and also they suppress cytotoxicity of the immune cells, which are important for the damage of the liver tissue. So as per Harrell's work, here we have a picture of the interplay between mesenchymal stem cells and the immune cells in the liver. So mesenchymal stem cells can, by producing these prostaglandins and immunosuppressive cytokines, immunomodulatory cytokines, they can shift the immune response toward the regulatory response with T regulatory cells, generation, also type 2 macrophages to shift the phenotype changes to type 2 macrophages, which are important for the reparation of the damaged tissue. Also, they secrete other factors such as vascular endothelial growth factor, hepatic growth factor, and others which work on hepatocytes. They prevent apoptosis, cell death of hepatocytes, and promote their survival and proliferation. And under specific conditions, they can also turn into hepatocyte-like cells, which is important for regenerative hepatology. So, due to their potent regulatory and immunomodulatory properties, these cells, mesenchymal stem cells, are considered to be potentially good agents in some liver disease, which are in which the immune response has an important role. And also some endogenous and exogenous factors also can induce the changes in mesenchymal stem cells, so they can shift to different phenotypes, and by their different phenotypes they can promote different outcomes in the immune response. So one of these exogenous hazards is cigarette smoke, and that's what we wanted to check in our study. Before, detrimental effects of cigarette smoke on survival and proliferation rate, also on migration and differentiation of mesenchymal stem cells have been demonstrated, but we wanted to demonstrate that in the murine model, in the animal model of acute liver failure, to see the role of these mesenchymal stem cells. We know that cigarette smoke impair synthesis of transcriptional factors that regulate the cell cycle, and also chemokine receptors, which are important for homing of stem cells to the inflamed tissue. So in our study, we used murine or mouse model of liver failure. We used alpha-galactosylceramide, which induces immune-mediated liver damage. And so we wanted to see in this animal model how the cigarette smoke would affect the outcome. So our aim was to determine the outcome, how cigarette smoke impacts the mesenchymal stem cells. So we sought to find differences in cytokine concentration, liver damage by transaminases and pathohistological analysis, and also we used flow cytometry to check the immune response or also phenotype of the immune cells. So first, for a generation of smoke conditioned medium, we used Borgwald machine. So a smoking machine with the puffing parameters, 35 milliliters was the liquid, and puff was every 60 seconds with a 2-second duration. For the generation of media for treatment of mesenchymal stem cells, this smoke from the machine was passed through a glass impinger and put into the mix of complete medium with fetal bovine serum for the cells. And was used, because we worked with cells, it was all aseptic conditions. For induction of hepatitis, we used alpha-galactosylceramide, and there were two groups, experimental and the control group. Control group only received saline, and experimental group received injection of this substance, which would induce liver damage by the immune response. And also the mice that were treated with alpha-galactosylceramide were divided into two experimental groups. One group would be given stem cells which were conditioned in complete media, and the other group would be given stem cells that were exposed to the cigarette smoke and media. So after the stem cells were cultured for 48 hours in the media, and then at the same time, when inducing the disease, this is acute disease, so they were given to mice. So our results have shown the differences in these three groups by every parameter, almost every parameter we sought. So the first, for the liver damage, as you can see, the control group or healthy group had significantly lower, because the liver wasn't damaged. But what's important here, that the group of mice that received, there was a difference between the two sick groups, the one that received mesenchymal stem cells and the other that received mesenchymal stem cells who were preconditioned in media which was exposed to cigarette smoke. Also, a histopathological analysis have shown that the necrotic tissue, coagulative necrosis, was extensive in the group that received just the alpha-galactylceramide, but there was a difference between that group and the group that received mesenchymal stem cells. So the liver damage in the group, histopathologically speaking, was lesser, to a lesser extent, in the group that received mesenchymal stem cells that were conditioned just in complete media. And on the other side, the mesenchymal stem cells that were treated with smoke, the histopathological damage was more severe. So next, what we wanted to here we have the analysis of histopathological damage. Next, we wanted to see the immune response. So we checked the concentration of inflammatory cytokines. So these cytokines, TNF alpha, interferon gamma, interleukin 6, are paramount in the liver damage in this model, and all of these cytokines were reduced. So concentration of these cytokines were reduced in the group which received mesenchymal stem cells, but in the group where mesenchymal stem cells were exposed to cigarette smoke, the difference was their protective, they lost their immunosuppressive and hepatoprotective properties. Here we also when we talk about the immune response, the interleukin 12 is important because it shifts the immune response to the detrimental to Th1 response and induces a production of hepatotoxic cytokines in a liver infiltrated with NK and NKT cells. So this here we can see that the production of TNF alpha and interferon gamma in NK and NKT cells were more pronounced in the group which received the conditioned stem cells, which were conditioned to the cigarette smoke and medium. These inflammatory, we checked the innate and also the adaptive immune response. In the innate immune response, NK and dendritic cells are most important, and these inflammatory cells were their number was significantly higher in the livers of animals that were treated with mesenchymal stem cells that were exposed to cigarette smoke compared to those which were treated with mesenchymal stem cells which were only treated with medium. Also, the higher presence of inflammatory interleukin 4, interleukin 17, producing NK and NKT cells. So these cells, NK and NKT, which produce interleukin 17, interferon gamma, TNF alpha, they are paramount for the damage in this model, and their numbers were higher in the livers of those mice that were treated with cells that were conditioned with smoke medium. So that confirmed our hypothesis that cigarette smoke impairs the ability of mesenchymal stem cells to suppress the inflammatory innate immune cells. Then we checked also the hepatotoxic T cells, so the adaptive immune response. And also the number of inflammatory TNF-alpha producing Th1 and interleukin-4 producing Th2 and interleukin-17 producing Th17 lymphocytes in the livers of the mice that were treated with media were smaller. So basically what we have here in the mice that received mesenchymal stem cells, which were conditioned only in media, they retained their immunosuppressive and hepatoprotective properties, while those who were treated with cigarette smoke, they lost those properties. So the immune response was more pronounced, more inflammatory by all parameters, both innate and adaptive immune response. I'm sorry because I'm taking much time. Also when we checked the immunosuppressive and immunoregulatory cytokines like interleukin-10, there were these cells, there was a significant increase in these regulatory cells in those mice that were given mesenchymal stem cells just in media. And also these immunosuppressive factors and hepatoprotective factors, the levels of these factors were significantly decreased in the supernatants of these stem cells that were exposed to the cigarette smoke compared to those that haven't been. That way, mesenchymal stem cells that were exposed to cigarette smoke couldn't suppress the production of these inflammatory cytokines, which then aggravated the clinical presentation and the molecular aspects of inflammation. And because this was the animal model, we also checked if this is true for the human T and NKT cells. These cells are the most important for damaging the hepatocytes. So we also we used the supernatant, so these secreted factors from the mesenchymal stem cells from the two groups, those that were just treated with media and that were treated with cigarette smoke, and we found that considerably lower levels of inflammatory and hepatotoxic cytokines in those cells that were cultured with mesenchymal stem cells that were not exposed to cigarette smoke compared to those exposed to cigarette smokes. And we have that both for T cells and NKT cells, and also CD4 cells, helper T lymphocytes, which promote inflammation. So to conclude, cigarette smoke has detrimental effects on immunosuppressive and hepatoprotective properties of mesenchymal stem cells. Exposition to whole smoke complete media induced generation of inflammatory phenotype in mesenchymal stem cells and reduced their immunoprotective or immunosuppressive and hepatoprotective capacity. And these cells that were treated with whole smoke complete media, they were not able to optimally produce immunoregulatory factors, so they failed to suppress T, NK, and NKT cell-driven liver inflammation, and they were incapable to promote repair and regeneration of injured livers in our animal model and in our in vitro studies. Thank you so much for your attention. It was a pleasure.